I want to welcome all of you to Pediatric Grand Rounds, and I will turn it over to our Vice Chair for Education, Dr. Sandrine Van Schaik, to introduce our speaker for today.
Welcome, everybody. It is my great pleasure to welcome our speaker today, Dr. Mark Walters. He's the Jordan Family Director of the Blood and Marrow Transplantation Program at UCSF Benioff Children's Hospital, Oakland, and Professor and Interim Chief for the Hematology Division in the Department of Pediatrics here at UCSF. He's Program Director of the California Institute of Regenerative Medicine Alpha Stem Cell Clinic at UCSF. And he received his AB with honors in genetics from the University of California, Berkeley, and his MD from the University of California, San Diego. So he's been in California for a long time, but then completed pediatric residency training at the University of Washington and hematology oncology fellowship training at the University of Washington and the Fred Hutchinson Cancer Research Center in Seattle. He was a junior faculty member in Seattle before matriculating to Oakland in 1999. And he's been active in cooperative clinical transplantation trials and has led several NIH-supported investigations of hematopoietic cell transplantation for sickle cell anemia and thalassemia. He has authored and co-authored a number of publications with a focus on hematopoietic cell transplantation for hemoglobin disorders, and he has a research interest in extending transplantation to young adults with hemoglobin disorders and other novel cellular therapies for non-malignant hematological disorders. Currently, research interests are focused on genomic editing and gene addition therapies as a strategy to extend curative therapy in all patients who inherit a clinically significant hemoglobinopathy. And we always ask our speakers, how did you get to be excited and passionate about this work? And his answer was that the intersection of basic science and hereditary hematological disorders with challenges of clinical care have provided strong motivation to investigate curative therapies for disorders like sickle cell disease. Extending these advances to an under-resourced, underrepresented group of patients is particularly rewarding. So we're really excited to hear about this work today, and I'll hand it over to Dr. Walter.
Thanks, Sandrine, for that kind introduction. So here's my title slide, Curative Therapies, Hemoglobin Disorders. I have these disclosures, but I don't believe any of these activities will relate to this talk today. So just by way of review, to remind you about the hemoglobin molecule, it's two alpha and beta chains, each of which contains a hemoiety that carries oxygen. And this molecule is uniquely suited to solubilize oxygen so it can transport oxygen to our vital organs, like the brain and the heart. And of course, the most important organ of all, the bone marrow. I have to put that plug in. So, but alterations in this molecule that alter its abundance or function and stability can lead to clinical consequences, including anemia, reliance on red blood cell transfusions, and in the case of sickle cell disease, a chronic illness. And together, mutations in either the alpha or beta globin chains account for the most common human genetic disorders worldwide, and that's because the trait condition confers a more likely and expedited clearance from the reticuloendothelial system in the case of malarial exposure. So even today, carrying a trait condition for a hemoglobin disorder generally extends resistance to that type of infection that's still quite common worldwide. Now, while there have been amazing advances in sickle cell disease supportive care, such that almost all children survive to the age of 20, this is still a very sobering observation. And this is a fairly recent publication by Mike DeBon and his colleagues in Vanderbilt and at the University of North Carolina, where beginning from age 20, those that make the age 20, he shows that the median survival in hemoglobin SS and S beta 0 thalassemia is still only in the 40s, mid 40s. And SC and S beta plus thalassemia, that genotype was generally creates a more favorable phenotype, has a very similar survival, mid 50s. So even with all of the advances and expansion of disease modifying therapies, this is a terrible disease with a 30-year decrement in lifespan compared to those African Americans who don't inherit sickle cell disease. So this is what we call a health disparity. So why is there a 30-year decrement? Well, a lot of it's related to the disease, but there's also been disproportionate funding levels over the years. And by way of comparison, cystic fibrosis that affects about 30,000 Americans compared with 100,000 Americans with sickle cell disease. This demographic is contrasted with the level of NIH funding. So they have nearly equal NIH funding despite the more prevalent sickle cell disease in this country. Cystic fibrosis, as we all know, is mainly inherited by those of Northern European descent. Sickle cell disease from Black and African American descendants from Africa and other parts of the world. And then foundation funding, the Sickle Cell Disease Foundation funding is really dwarfed. And so when you take into account all the funding for research that exists for these two disorders, there's an order of magnitude less funding for sickle cell disease. So this is what I would call an example of structural racism, and how this kind of lack of attention and resources devoted to curative therapies for sickle cell disease has contributed to I would argue to the disparate outcomes in terms of lifespan that I've just showed. So this is an important feature of the disease that we continue to struggle with, particularly since the hallmark of the disease, which is pain, is not a symptom you can verify by a blood test. As everyone knows, one has to either believe or not believe the report of pain. And because pain treated by opioids has a certain stigma attached to it, it can lead to ineffective treatment or a delay in treatment, which is again a struggle for our families and patients affected by these diseases. So that's the rationale for pursuing a curative therapy. And I embarked upon this journey, 30 years ago, when we did our first clinical trial, multi-center trial, showing that allogeneic hematopoietic cell transplantation using generally bone marrow as a source of cells, but also cord blood and mobilized blood stem cells is curative for this disorder. And in fact, it works in the majority of the time. I'll show you the data. But unfortunately, very few individuals who inherit sickle cell disease will have a well-matched donor and can take advantage of this curative outcome. So gene addition therapy using autologous cells, so a person's own stem cells, whereby an anti-sickling beta globin or the gamma globin gene, which has potent anti-sickling activity, can be applied. Gene addition therapy to induce fetal hemoglobin, gene editing for gamma globin expression in autologous stem cell transplantation. And finally, the project that I've been working on locally, gene editing for sickle allele correction to mitigate for disease mitigation in autologous cells. What I won't be talking about, but something that's in the pipeline in a remarkable collaboration we have with the Innovative Genomics Institute, Jennifer Doudna's operation at UC Berkeley, is to deliver by antibody targeting the gene editing reagents directly into the marrow where the stem cells reside, and to do this in the absence of any sort of chemotherapy or radiation to ablate stem cells. Now, we're not anywhere near turning that into a clinical trial, but it's something that I wouldn't be surprised if we make a lot of progress in the next several years. So what are the results of bone marrow transplantation for sickle cell disease? This is the gold standard, and these are Kaplan-Meier probability curves, and you can see that the red curve, these are the youngest children who have an HLA identical sibling donor. The cure rate or the event-free survival probability is around 90 to 95%. So really quite good result. These are retrospective transplant data, a training cohort and then a validation cohort, 1,500 transplants in total, divided by 1,000 in the training cohort, of 500 in the validation cohort. And then I draw your attention to the lower green curve, which shows a inadequate outcome, which is an event-free survival probability that settles out around 53%. And these are all these are the patients who are older than 12, so not that old, 13 years of age and older, who've received either a mismatched related donor, mismatched unrelated donor, or even a matched unrelated donor transplant. So the middle curve are the older patients who've received an HLA identical sibling donor. So since most patients lack an HLA identical sibling donor, we're really going to focus on the green curve. So these are individuals who none of us would really have a lot of confidence in taking to transplant with a 50% event-free survival probability. So the other principle, so that's the rationale for considering gene therapy, either gene editing or gene addition therapy using autologous cells, because everyone can act as their own donor. But the other important principle is something we observed early on in the allogeneic transplant setting for both thalassemia and sickle cell disease. And so even though we delivered ablative chemotherapy to make room for the donor cells to engraft, serendipitously, sometimes as little as 20% of a patient's marrow after a transplant was made up of the healthy donor cells. And what was surprising about that was that that's actually sufficient to cure the disease. So this phenomenon occurs because the healthy donor erythrocytes and erythrocyte precursors in the marrow outlast the sickle cell disease and beta thalassemia endogenous cells. And so they're naturally enriched. We don't have to give any sort of selection. And it indicates that in a gene therapy, gene editing protocol, if we could correct at least one sickle mutation in at least 20% of the engrafted stem cells, that might be curative, or certainly would have a significant clinical benefit. So that's our target, and we'll be referring back to this phenomenon later. So another important consideration is that if we're going to introduce an anti-sickling hemoglobin, and these are data, a slide from the Bluebird Bio-sponsored trial, the results of which were just published last month, the largest trial performed to date, has to do with the distribution of the circulating of the anti-sickling hemoglobin in the circulating red cells. And so antibodies that fluoresce were directed at the sickle hemoglobin, the healthy hemoglobin A, and then the gene therapy hemoglobin that's termed beta A T87Q. So there's been a single amino acid swap at position 87 such that it mimics fetal hemoglobin and has strong anti-sickling activity. So in a healthy person with normal red blood cells, then this orange indicates that all of the red cells have all normal hemoglobin. Of course, those won't undergo any sickling under hypoxic conditions. And this contrasts with a person who's inherited sickle cell anemia with 2 beta S alleles, and they only make hemoglobin S. And so under hypoxic conditions, they will sickle and interfere with circulation, et cetera, et cetera. Now, what we sometimes do to lessen the occurrence of severe vaso-occlusive events is to put persons on a transfusions who have sickle cell disease. And what we achieve here is a mixture of cells that are all hemoglobin S and all hemoglobin A. And in this situation, unfortunately, those cells that still retain 100% S will undergo sickling under the right conditions. Unlike sickle cell trait, where you see the stippled pattern, where all the cells have a mixture of hemoglobin A and hemoglobin S, and they don't sickle at all. And so what we asked in the gene therapy trial is the pattern of that of stippling, where most of the cells contain at least some of the anti-sickling hemoglobin, or is it more like a person on red blood cell transfusions? And so the answer is stippling. So this is a cohort of patients, about 35 patients who underwent the gene therapy treatment with sickle cell disease, and shortly after transplant, by 3 to 6 months, you see this emergence of the stippling pattern, and the red blood cells with only hemoglobin A go away because the transfusions are stopped. And at 2 years, a small population, about 10 to 15% of the cells, still retain 100% hemoglobin S. But we would predict this pan-cellular distribution of the anti-sickling hemoglobin should be sufficient to prevent clinical complications. Now, in thalassemia, the target is a little bit different, which is that instead of having red blood cells that last longer in circulation with an anti-sickling hemoglobin that interferes with the principal manifestation of the disease, which is pain from vaso-occlusion, the problem in thalassemia is that of ineffective erythropoiesis. And so what we show here in a couple of patients who've had a successful outcome, that is they got the gene therapy hemoglobin. It's actually the same vector that we use in sickle cell disease. And over time, the transfusions were stopped. But despite that, while the cellularity of the marrow improved, the MDE ratio, which should be 3 to 1, still remains inverted. So one of the issues is that we see this phenotypic correction of the thalassemia in the transfusions are stopped after the successful gene therapy treatment, but there's still signs of the underlying disease because there's still ongoing ineffective erythropoiesis. And over the long term, there'll be this question of, well, what's the impact? Is this truly curative, or is this just another disease modifying approach? So one of the things we have to do in all of these trials is define what we mean by event-free survival and measure its durability. So we focused on assessing the impact of the therapy on the biology of the red blood cell. So hemolysis and ineffective erythropoiesis, for example. We assess the impact of therapy on its clinical expression. So is pain eliminated? Is there stabilization of end organ function and protection of organs like the brain and the lungs and the liver? And then assess the significant adverse outcomes. So in an allogeneic transplant, the one that we're trying to avoid is chronic graft versus host disease because it has a strong impact on the quality of life. But there are all these other risks, including treatment-related malignancy, that have to be assessed. And when we're doing genomic targeting and gene replacement, what are the benchmarks? What fraction of the stem cells have to be corrected or targeted, and is that durable in the long term? So with the FDA, we had a workshops a couple of years ago with the American Society of Hematology to try to establish some critical endpoints that we would employ globally across all the gene therapy and gene editing trials. And this is just one example. So if the endpoint is anemia and hemolysis, we should measure hemoglobin, reticulocyte counts, LDH, hemopexin, haptoglobin, et cetera, et cetera. And for erythropoiesis, there are markers of ineffective erythropoiesis, such as the soluble transferrin receptor erythropoietin, and then the appearance of the marrow under the microscope. And then we might even see a gradation of the clinical effect. So a cure would be all of these parameters normalized, and then obviously a failure would be no improvement. But what if we end up in the middle where there's an improved anemia, but ongoing hemolysis, or improved ineffective erythropoiesis, but not resolved? So these are all things that we consider in study designs in terms of how we would assess the success or lack of success of our particular intervention. Now what about pain? I mentioned it's the hallmark of this disease, and again, our view of this is improving over time. And this was a remarkable study was published just last year, having to do with the evaluation of longitudinal pain in adults with sickle cell disease. So a 6-month study, 37 patients and about an equal number of controls were analyzed. And what was remarkable about it is this pie chart divides pain into vaso-occlusive events that have a beginning and an end, and then within an event, vaso-occlusive days. And what's remarkable about it is that most patients have learned that they can treat pain really better at home than they can in the hospital. So hospitalizations were only sought about 13 to 18% of the time, and even direct healthcare utilization wasn't always sought. So about a third of the time, vaso-occlusive events and these vaso-occlusive days are handled at home. And again, this has to do with the type of care that across the country is delivered in the emergency department setting, where the report of pain might or might not be believed. The institution of opioid therapy may or may not occur. And there's a lot of stigma attached to that asking for pain relief, which could be interpreted as drug-seeking behavior. So again, how is pain ranked at home? So this is a pain score and then a functional score. So a pain score, the higher score means more pain, and the red bars indicate the days when a person in the survey wasn't experiencing pain or wasn't experiencing a vaso-occlusive day. And then the green is vaso-occlusive days treated at home, and then blue is vaso-occlusive days treated in the hospital. And there's really no difference, very little difference between pain treated at home and in the hospital in terms of its score. The worst, the least, average, and right now it's shown. And then as far as physical functioning, social functioning, again, it's surprising and interesting that the functioning level for pain treated at home is really not terribly different than the level of functioning for days without pain. these are really resilient individuals who've learned how to function at a very high level, despite what would ordinarily be debilitating level of pain. And the tiredness score is also shown on the right. Again, it is worse on vaso-occlusive days, but not particularly good on non-vaso-occlusive days. So this is one way of assessing pain using a patient-reported outcome approach. That's different than the way we measure it in a gene therapy trial, for example. And then one of the things that happened in the trial was to show that when they had a vaso-occlusive event, a blood sample was drawn, and this shows quite conclusively that the biomarkers of inflammation were increased during vaso-occlusive episodes. So C-reactive protein and interleukin-6 in particular were elevated. So this is a remarkable result from the Bluebird Bio-sponsored gene therapy trial that was just published last month. 35 patients in total were treated with the gene therapy vector. And this swim lane chart just shows vaso-occlusive episodes by those blue diamonds before, in the 2 years before, and then up through 36 months post-infusion. And you see this remarkable absence of severe vaso-occlusive events culminating in a hospitalization after the infusion. And so there was a lot of celebration about this, appropriately, because it's a spectacular result. But the question is, are we really capturing all the pain that occurs in sickle cell disease when we assign the definition as it was assigned in this study? And the possibility that this may take some time to resolve was suggested by this study. This is a bone marrow transplant study conducted by Matt Shea and his colleagues at the NIH, enrolled about 36 patients. And what he showed on the left is that the hospitalization rate compared to before this HLA identical sibling bone marrow transplant declined significantly in the 3 years post-transplant. But look at the morphine equivalence doses as these evolved over time in the weeks after transplant. And so initially, opioid doses were quite elevated, but it took up to 18 to 20 weeks before opioids were discontinued continually. So there is this high impact chronic pain of sickle cell disease that has a nociceptive component that takes a long time even after a successful intervention like this one to remodel. And so again, patient expectations when they go into a bone marrow transplant have to be modulated appropriately. So just because healthy donor hematopoiesis has occurred by 8 to 10 weeks after transplant doesn't mean the pain will necessarily be gone. And this is something that we struggle with in all of our patients. And this is a busy table, but it amplifies the point, which is, so these are about the same number of patients, 36 patients who were enrolled in that previous trial, organized into these 4 columns: those who had acute episodic pain only, and then 3 different categories of chronic pain. And what's fascinating is that the episodic pain only group had that resolved post-transplant, by 1 year post-transplant. But the chronic pain of these various subtypes, some of which is neuropathic pain, was not as successful. So in some cases, 50 to 80% of the patients were still receiving short-acting opioids 1 year after transplant, and some were still being hospitalized for pain. So how do we define cure? Well, resolution of pain, both in the inpatient outpatient setting, may not be appropriate for all the patients. And so this is another thing that we try to build into our study design. And finally, I'd just like to point to this analysis using patient-reported outcome measurement called PROMIS 57 in the Bluebird Bio-sponsored gene therapy trial. So this was conducted in 25 of those 35 patients who ultimately were treated with the gene therapy drug product. And the patient-reported outcomes were sorted into 2 strata: those who had baseline scores near or better than the population norm, and those patients who had baseline scores in orange worse than the population norm. So a 5-point or greater point change in those domains is considered significant, and in the pain intensity NRS, a 2-point or higher change is considered significant. And understanding, of course, that this instrument doesn't account for all the environmental and contextual factors that influence a patient's report. So these are data tracked over 24 months post-infusion. This is the physical function domain from PROMIS 57. And again, the orange are those who have a worse than baseline, defined as 50, worse than population norm at baseline, and then those who were at the population norm. And both groups improved over time. In the black curve, these dots represent the total group. So not surprisingly, physical function improved over 24 months, and it was stable. Pain interference. So in this case, improvement moves the curve lower, so there's less interference with physical functioning, and both groups, again, those better than and worse than the population norm, saw improvements over time that was stable. So 56% of the patients at month 24 had a sustained improvement in their pain interference score. So this has also been performed in bone marrow transplant studies. So this is a study that was published 5 or 6 years ago, a small number of adult patients. And what's interesting is that in some of these domains, like vitality and general health, there's quite a strong treatment effect. But in other domains like physical functioning, mental health, emotional role, there was no statistically significant difference. So according to patient-reported outcomes, there seems to be a clustering around a certain subset of these domains where there is improvement, in other domains, particularly in the mental health and social role functioning, where the improvements aren't necessarily as obvious or occurring. This is another group, patients, these are children less than 21 years of age who had a busulfan-based transplant or a reduced intensity type transplant. That's the way we prepare patients for a bone marrow transplant, and was compared to children with sickle cell disease who only had the consultation and didn't undergo the transplant. And a broader array of health-related quality of life instruments was used in this particular study. And what it showed, surprisingly, these are the 3 instruments that were tested. Compared to controls, really not much of a treatment effect across these domains. But as far as healthcare utilization, a significant improvement in the group of patients who went on to get a transplant compared to the controls who didn't, and that was highly significant. So health care utilization certainly improves after a successful transplant, but these quality of life domains, it's not clear to us exactly why that's more elusive. And this is a study that we did this was a collaboration with Lakshmana and Krishnamurti at Emory University, and we enrolled about 20 adults in a pilot investigation of bone marrow transplantation, and showed that the pain interference scores over 6 to 12 months after the transplants improved. The fatigue score got better, physical functioning improved, but these other domains, satisfaction, sleep disturbance, anxiety, and depression, were not necessarily improved. And so some of this has to do with the frame of mind that the patients have. They have sickle cell disease, that's their identity. And if we don't prepare them properly with what is your view after we've eliminated the sickle cell disease, these problems with pain and anxiety that existed before the transplant won't necessarily go away without intervention. And the last thing I wanted to show, one of the important ways that we prevent stroke in children with sickle cell disease is to screen them with a transcranial Doppler ultrasound. And those individuals who have high velocity in their cerebral arteries are offered red cell transfusions, or in some cases hydroxyurea. And this is just dramatic data showing that the risk of stroke was significantly reduced after instituting transfusions. And so this is an established therapy. So Françoise Bernaudin and her colleagues in France did a comparison cohort study where patients who had an HLA identical sibling donor went to transplant, and those who lacked a donor didn't. And she followed their transcranial Doppler ultrasound velocities, which improved after a transplant, shown here in this upper panel, and which were stable for those receiving regular transfusions. And then also assigned an MRI stenosis score, which also improved after transplant, but was stable in the standard care group. So these are the standard care groups showing stabilization. So it seems that the neurovascular disease of sickle cell disease in childhood might also be successfully modified after these type of interventions. And finally, this shows neurocognitive data, and these are 13 participants in an unrelated donor bone marrow transplant trial in children, comparing before and after full-scale IQ, verbal IQ, PIQ scores, and mostly these remain stable as expected. We wouldn't expect to see much of an improvement. And if there is an improvement, it tends to be because they attend school more often and have less absences and are able to concentrate better when they are learning. So that's, I think, the principal benefit in neurocognitive outcomes after a transplant. So so this reminds me that we're gonna go back to this question of curative therapies. Can we establish a cure, and then over time is it durable? And so what we've built into these studies is up to 15 years of follow-up. This is mandated by the FDA, but we're all terribly interested in defining this. And so in addition to so this is the vector copy number on target editing fraction that can be monitored in the peripheral blood over time, and then blood transfusion events, the blood counts, and the type of hemoglobin they're making. All of these will be tracked over time to assess the efficacy and durability of the clinical benefit. Now, one of the things that has occurred in the course of this gene therapy trial sponsored by Bluebird Bio is the observation that two of the now in total 42 patients who were treated developed acute myelogenous leukemia. And these were two individuals who were enrolled in the trial when we were using bone marrow in lieu of mobilized blood stem cells as a source of their stem cells for the lentiviral vector transduction. The first was a 45-year-old who developed myelodysplastic syndrome with monosomy 7, and when the transformed to leukemia also had driver mutations, RUNX1, KRAS, and PTPN11 that were associated with this event. After he transformed to leukemia, the leukemic blasts were assessed for the presence of the vector, and there was no vector in those blasts, indicating that this leukemia arose from residual host hematopoiesis and was not a genotoxic event, suggesting that the possibility that exposure to busulfan, which is an alkylating agent, triggered this treatment-related leukemia. A second patient who also received bone marrow also developed monosomy 7 with RUNX1 and PTPN11 driver mutations. And in this case, the vector was detected in the leukemic blast population, and the vector had integrated near a gene called VAMP4, which is just a membrane protein housekeeping gene, not associated with AML, and probably not causal. So in this case, there may have been something about how we select and expand and then repopulate the hematopoietic stem cells in sickle cell disease. Somewhere in that journey, the leukemic transformation occurred. So there are some hypotheses about what might be going on here, because this is a significantly increased risk compared to the healthy population that we've considered. So the marrow environment in sickle cell disease is inflammatory. It is a milieu with stress erythropoiesis due to high demands from the hemolytic anemia. And then from this milieu, we extract stem cells trying to proliferate under these conditions, and then they're expanded in vitro to select for cells after transduction that might select for cells with pre-existing mutations that favor proliferation or survival under those in vitro culture conditions. And then, of course, the transduction itself may trigger a malignant outcome by genotoxicity, by activating an oncogene. And finally, transplantation of a relatively small number of these repopulating stem cells into an ablated host, again, causes proliferative stress selected for cells that may have pre-existing mutations that favor proliferation or survival. And then I've already mentioned that the exposure to busulfan may increase the mutational burden on any residual host cells. So the sponsor, Bluebird Bio, came up with this slide, which I think is interesting. The group A patients, not only do they use bone marrow, which has a smaller number of stem cells that will be transduced than the peripheral blood stem cells used in group C, they also had a manufacturing protocol that was less efficient, only transduced about 30% of the stem cells, compared with the revised, improved manufacturing protocol where 70 to 80% of the stem cells were transduced. And so this led to a high median unique insertion sites with reduced proliferative and hematopoietic stresses, according to this hypothesis, compared to the initial iteration where there was high proliferative hematopoietic stress due to and low median unique insertion sites. So this also suggests a pathway to perhaps mitigate the risk. So where are we with this risk of hematological malignancy in sickle cell disease? So we've decided to assess for insertional mutagenesis after gene addition therapy by viral vectors. We also are conducting assessments for pre-existing MDS, myelodysplastic syndrome, with variant allele or driver mutations in clonal hematopoiesis before enrollment that would exclude a patient from proceeding to the transplant. And then should we be screening using molecular means for high-risk clonal variants, clonal hematopoiesis in a diagnostics panel? The answer is yes, we're proceeding to do that. And then, of course, post-transplant monitoring for clonal hematopoiesis. So I hope in this part of the talk, I still have a few minutes left. I've imparted some of the important factors that need to be considered. Is the effect durable, and is the magnitude of clinical and hematopoietic outcomes? This would require long-term monitoring. Incomplete correction of the genotype is a very likely outcome, but we rely on that mixed chimerism principle to have a clinical benefit. Patient-reported outcomes are being used more broadly and perhaps give a better picture of what we mean by cure. And then these cardiopulmonary outcomes after curative therapies is really incomplete and needs to be studied. So I want to finish with two clinical trials that we're about to open at UCSF at the Oakland Children's Hospital. This one's led by a team at Boston Children's. David Williams and Erica Ezrick led the phase 1 study. This is going on to a phase 3 licensing trial. And it builds on this observation that a DNA binding complex protein called BCL11A is really the master regulator of globin switching that happens at birth. And when it's expressed at birth, it silences the gamma globin gene by binding the gamma globin promoter and allows beta globin to be made. Now, I've already said that fetal hemoglobin has this strong anti-sickling effect. And in this mouse model of sickle cell disease, where the blood smear shows these classic irreversibly sickled cells, this is corrected when the BCL11A is knocked out and the fetal hemoglobin is awakened in this mouse model of sickle cell disease. So the team at Boston Children's has created this vector. It has erythroid regulatory elements driving expression of a BCL11A targeting sequence, a microRNA derived scaffold that, when it's expressed, suppresses BCL11A just in the erythroid lineage. And that's important because this gene its expression is necessary for hematopoietic stem cell and B lymphocyte development and maintenance. And treating 6 patients, they showed very nicely this hemoglobin F induction in the 30 to 40% range, And then the F cells in circulation, these are fetal cells that have a high level of fetal hemoglobin. They largely replace all of the cells in circulation. 70 to 80% of the cells are F cells, and that translated into a strong clinical benefit and a marked improvement in the hemoglobin, with a level of fetal hemoglobin per F cell where 10 is the target that mimics hereditary persistence of fetal hemoglobin, which when patients inherit the sickle allele with HPFH, the phenotype is sickle cell trait. So a very nice proof of principle. And now we're going to go on to a 25 patient licensing trial, and we'll be co-leading this trial using busulfan as a single agent in preparation, followed by infusion of the lentiviral gene modified cells that express this microRNA that suppresses post-transcriptionally BCL11A production. So to be eligible, patients have to be between 12 and 40 years of age and to have at least 4 vasoclusive events in the 2 years before enrollment and adequate hematopoietic stem cell function. The other trial I wanted to describe, which is transitioning from the preclinical to the clinical trial phase, is our own curing sickle cell disease with CRISPR-Cas9 genome editing trial. This is the amazing project team that contributes. At the IGI, Mark DeWitt was our program manager for the preclinical phase. Don Cohn and his colleagues at UCLA run the CMC for GMP manufacturing. The IGI program is led by Fyodor Urnov now, and David Martin and Wendy Megas did a lot of the preclinical mouse studies at Corey. So this is, I know, a little wonky, but this is this shows the components of the gene editing system by CRISPR. So there's a guide RNA that leads the Cas9 nuclease to the sickle mutation, and so this is where the double-stranded break occurs in the DNA. It's some bases downstream from the actual sickle mutation. So this template, which is a single-stranded oligonucleotide, directs correction of the valine for glutamic acid swap that would cause correction. And so unfortunately, not very often, but sometimes the replication fork falls off before it reaches the sickle mutation. So you do correct this PAM mutation. I mean, you do insert this PAM mutation, which you need to do so you won't re-edit the guys you've already corrected. And it gives us a nice control when we look at enrichment of the corrected cells in the red cell population. So this just shows that the targeting is both efficient and there are limited off targets. As it turns out, there were only 2 when we used the wild type Cas9, and then since we switched to the high fidelity Cas9, there's only off targeting at one site, and it occurs about 4% of the time. And so this is shown in this slide, where when we use the high fidelity Cas9, we lose the off target site 2, and this is a single off target that occurs in chromosome 9 in a gene desert, so it doesn't cause any issues at that particular site. And then in addition, what we show with a drop of digital PCR assay, looking for chromosomal translocations between HBB, which is the beta globin gene, and this off-target site 1, there's a way to create these nested primers to detect for that. And this just shows the assay that Mark DeWitt worked out to show that it was had a sensitivity down to about 0.01%. Using the manufactured lots that we generated, the translocations were at or below the level of detection. Some of these are mock runs, so we don't think there's any translocation, at least to the level of sensitivity of our assay. So that's also very important. But one thing we worry about is this phenomenon called Chromothripsis, and this is interesting. The same reagents that have been used in an ongoing CRISPR therapeutics trial using CRISPR technology to target the BCL11A gene, erythroid-specific enhancer. And what happens in 2.5% of the cells are these fusions that occur where the chromosome completely rearranges after the gene editing, which is something that we're also concerned about in our own trial. So the genomic toxicity, if you will, and the ability to detect it, we're still in the early stages. So that's why these phase 1 studies are both exciting and worrisome, because there are toxicities that we may or may not be able to observe unless there's a clinical outcome. So the process is diagrammed here. We begin with stem cells that we mobilize from sickle cell disease patients. The gene editing reagents, the oligonucleotide, the guide RNA with the Cas9 are electroporated into the stem cells, and then at the manufacturing site, these cells are cryopreserved. They're analyzed for QC, for level of editing and viability and so forth and so on, sterility. And then if they meet the quality release criteria, they're shipped back to Oakland, where we infuse them after busulfan myeloablation. And we've done 6 clinical runs, 6 manufacturing runs, 3 of which were at clinical scale. And what we see is that we correct a single allele 20 to 25% of the time. But then another thing that happens that will also have to be mapped carefully is we also have these insertion deletions that introduce a beta thalassemia allele. So we believe that with 20 to 25% editing, these thalassemia alleles won't contribute to erythropoiesis, and the corrected healthy cells will expand and overcome the effects of the indels, but that's something that we can only test in a clinical trial. The proof of that is shown here when we take the marrow samples and create colonies and pick the colonies. We show that the way the corrected allele segregates, about half, 40%, half of the stem cells are corrected, the rest being uncorrected, and then about a third of the time this insertion deletion distribution. And finally, the last slide that I'd like to show is this one. Again, kind of wonky, but what we've done here in the mouse xenografts is that we've harvested marrow from the mice and then sorted the human cells either as whole marrow CD34 positive stem cells or CD235A positive erythroid precursors. In the unedited, there's no difference across those lineages. And when we have that control where we've corrected the PAM, but we haven't extended the correction to the sickle mutation, no enrichment across the lineages. But look what happens when you correct the sickle mutation. You see this strong correction accounting for 50% of the colonies, of the mice, I should say, having a corrected allele in the erythroid population. So this type of enrichment at 20 to 25% correction in the marrow and stem cell is exactly what we want to see. And then when we've introduced a non-homologous end joining insertion deletion out of frame, those red cells drop out of the equation because they cause a thalassemia and they don't contribute to erythropoiesis. So everything that we would predict is shown in this figure, and it's one of the preclinical studies that convinced the FDA to give our IND a clear to proceed notification. So in summary, patients, families, referring physicians have concerns about myeloablation and associated toxicity like AML. So we'll have to spend a lot of time counseling and preparing our families and adults who decide to proceed about these risks. And we're also hampered a bit by not having 10 and 20-year long-term follow-up data. So, and this is concerning because there was a patient with thalassemia who had to resume transfusions after 10 years after a successful gene therapy treatment. Is there a risk of clonal hematopoiesis, selection, proliferative clones, AML? That's something that we'll have to learn. And finally, maybe most importantly, if we establish this as an effective and safe treatment, will there be access to it? And how do we establish broad versus limited availability? And that health policy issue is going to be a terribly important one to focus on. Not after we get approval or one of these gets approval, but right away. So I just want to acknowledge the funding agencies that supported this work, and I appreciate your attention.
Thank you so much for a really great talk. There's a number of questions in the chat, so I'm going to start from the first one. and hopefully we'll get through all of them. So Dr. Jim Bear asks, do severity of disease or duration of symptoms predict impaired resolution of chronic pain post-transplant? And then an immediate follow-up question, are there other predictors of persistent pain?
Yeah, I can share our experience, which is that if you take a 3 or 4 or 5-year-old to transplant, because they have just have had that acute episodic pain, that resolves completely after transplant. But what we're observing in the adults who have this, what we call high impact chronic pain, they have pain on most, if not all days, treated by opiates. That type of pain, which is again, it's nociceptive and it has a chronic, it just doesn't resolve quickly. And so what we have to do is have a plan for tapering the treatment and multimodal therapies that address also pain, anxiety and depression, and incorporate things like massage and acupuncture as we can to make that transition. And it can be very disappointing. So, but is there a test or is there a characterization that we can use to predict that in every case? I'm afraid there isn't.
And then there's a comment and question by Dr. Matthew Kahn, who says, beautiful and inspiring work. There have recently been findings that Cas9-induced double-strand breaks cause loss of distal arm of the chromosome. Is that something that you have looked at for the Cas9 approach?
Yeah, we did, and we used a device called SAGE, which is a way for you to load the whole chromosome in a very large gel. And surprisingly, we have not seen long-range deletions or rearrangements. But the other thing in our favor is that the beta-globin gene is very near the distal telomere. So even if you lop it off, there's not much beyond it that you would lose. So we have that serendipitous advantage, unlike the BCL11A gene, which on chromosome 6 is near the centromere. So there's lots more mischief that could occur there. But yeah, we have a whole separate grant award from the NIH to look at these long range, unbiased looks for large deletions, rearrangements, insertions that may be occurring, because once we target the on target and the off target, that's a PCR amplification and it's biased. So we also need an unbiased look. So that's a good point.
And then Dr. Efraat Lelkes says, thanks so much for this excellent grant rounds and for your work. The continued pain and quality functionality scores, despite improvement in the underlying disease in your sickle cell patients, is curious, though perhaps not surprising considering their chronic illness, and perhaps, as you mentioned, systemic racism. Are psychosocial resources, including pain psychologists, available for your patients and families?
Yeah, we do have a remarkable team. We have a anesthesiologist, Steve Long, who has partnered with us to do assessments in all of our adult patients, in particular, set up pain plans that the patients can participate in, and then he follows them after the infusions to help us with the reduction in the pain. What also happens in sickle cell disease, I didn't mention this, is that any painful stimula is massively amplified. So the mucositis, which is common, and virtually all patients get it after busulfan, its duration and severity is just so much worse in these sickle cell patients than some of our others. And so that needs careful management by pain management team. And the anxiety, the counseling, the cognitive-based therapy, that's also terribly important. Now, not everyone that we've treated is interested in having the multimodal approach, but we certainly offer it, and I would like to expand that palliative care team approach using multimodal therapies in our patients. And I think we do a pretty good job, but there's definitely room to expand that, especially if this becomes a more common form of therapy.
And then Dr. Brian Feldman asked, does having mosaic hemoglobin increase or decrease the risk of an immune response to the corrected non-self hemoglobin?
Yeah, I've wondered about this over the years, and I don't know, it's because the red cells are anucleate. We just don't see immune responses to the novel hemoglobins, either in the gene therapy or after an allogeneic transplant. So that doesn't mean it couldn't occur, but it hasn't been observed so far. So there's no class 1 for a dendritic cell to interact with.
And then the last question is from Dr. Elliot Wyszynski, and I'm not totally sure if I understand it, but I'm going to read it out loud, and then hopefully you know what he is after. He says 50% decrease pain transfusion, improved cognitive impact. It affects improve when anti-P selection of others approved drugs. The comparison of gene therapy should be compared to modern therapy.
Well, yeah, who could argue with that, Elliot? I mean, what I'm talking about is a invasive, intensive therapy that only a few families and patients are willing to take on, at least in its current form. And the vast majority of patients will rely on these disease-modifying therapies, which, as you point out, do carry benefit. But the rationale is, I think you would agree, a good one, which is that given the choice between taking a medication for the rest of your life, which you may or may not respond to, hopefully you would, as opposed to a one-time curative therapy, there's benefits to consider that might be compelling for a one-time curative therapy.
I think that brings us to the end of our questions.
Great. Well, they were wonderful questions. Thank you for your attention and for listening in today.
Well, thank you, Mark. That was really fabulous. I want to compliment you and your whole team on just an amazing program you have and the progress you've made. It's really transformative to the lives of these children and adults. So thank you, and thanks to everybody for participating, and we'll see you all at the next Grand Rounds.
Bye.